Jacob, and as the first recipient, myself and my colleagues in the lab, of course, have the greatest debt of gratitude to the Beutler family, the Beutler Foundation, to Bruce, not only for the financial support that's enabled the work, but friendship and advice, scientific advice, collegiality, and I think I won't repeat all of the thanks to everyone involved, but Bruce did mention Jacob, who really, with his connection with Bonnie, Bruce's mother, who I had the privilege of meeting and knowing and visiting in La Jolla a few times, has shepherded this success story all along. So thank you, and appreciate it very much. And people in the lab are here, and I'll show them and thank them at the end with some pictures. So you heard the title, and the challenge that we're facing or trying to address is one aspect of a global disease challenge, and that is kidney disease. End-stage kidney disease affects around 12 million people worldwide. Only a subset of them are actually treated with dialysis or transplantation, and many aren't able to get that treatment. And for any patient with end-stage kidney disease, there are some 50 with earlier stages of chronic kidney disease. And this is not a success story that we're preventing end-stage kidney disease. It's actually a non-success story. That is, the majority of chronic kidney disease patients actually succumb to complications, mostly cardiovascular, before progressing to end-stage kidney disease. And in chronic kidney disease, there's probably a 10 to 15-fold greater age-adjusted non-renal morbidity and mortality compared to the general population. So it is everyone who's spoken here, almost everyone has tried to play up the importance of the particular disease challenge that they're addressing. Now, if we're in a genome study mode, we need to think about whether there is a heritability component. How heritable is what we're trying to examine. And if you look overall at chronic kidney disease, then for children, and we have pediatric nephrologists here, obviously the genetic contribution is rather great, at least 50%, much of it monogenic with high penetrance Mendelian inheritance. In adults, it's around perhaps 20%, some of it polygenic or oligogenic, but surprisingly recent information is showing that there's a relatively high contribution of single gene mutations with variable penetrance and pleiotropic clinical presentation. So sometimes a particular gene, which is always thought of giving a particular presentations, such as an Alport syndrome gene, actually looks like nephrotic syndrome or some other form of kidney disease. And I think a lot more attention is now going to be paid to single gene causes of what look like pleiotropic chronic kidney disease disorders, much going together with what Professor Beutler showed this morning. And now it's becoming a standard approach to consider whole exome sequencing as an early step in the investigation of chronic kidney disease of unproven cause, certainly in children and even in adults. Now, so there is a clear heritable component and an important approach in this regard. And what about treating chronic kidney disease? Well, it's been pretty stagnant for many, many years with non-specific approaches that address perhaps glomerular hemodynamics, perhaps scarring, and so forth. And part of the complacency has been the thought that dialysis and transplantation can save lives, but we see that there's a much, much bigger problem here. And therefore, the time has come to look at more specific and effective therapy, and it seems that it comes together with the realization that perhaps by discovering targets based on genetics, we might be able to get that way. And we're focusing, as I've spoken about twice previously, as a first recipient. By the way, as the last speaker, I also want to thank the audience for sticking around and staying, because that's also not trivial. We have been focusing, as I've spoken about, on a particular subset of kidney disease, which covers a large number of individuals, and that is kidney disease that is responsible for the fourfold approximately increased risk in people of sub-Saharan African ancestry. And about 70% of that increased risk is attributable to causal variants at one genomic locus called which is the gene ApoL1 encoding apolipoprotein L1. I've spoken about how that population-based gene discovery was made a number of years ago by our group with Shai Tzur and many others who are sitting here, whose pictures I'll show later, and concomitantly by a group in Boston, Giulio Genovese and Martin Pollack's group. And since that time, there have been close to 200 publications by about 5 or 6 hyperactive, hypercompetitive groups around the world, both at the level of genetic epidemiology, mechanism, and some implications already for clinical management and therapy, and we've been lucky to be continuing to work on that, and a lot of that ability comes from the support that we get from the Beutler grant over the years. So I'll share some of that with you, and again, I won't go over how the discovery was made. It involved a form of what's called admixture disequilibrium linkage mapping, but the gene this just shows the domain structure, and it was actually studied long before any understanding of an association with kidney disease in the context of infectious disease as part of the innate immune system in terms of a circulating gene product made by the liver that protects humans and subhuman primates, but not most species, against certain forms of trypanosomal parasitic disease, which can be lethal. The ancestral form, which was given the name G0, is what most people have, and in certain regions of sub-Saharan Africa, because of the evolution by pathogens, just as we've heard, but this time parasites, of ways to resist the endogenous innate immunity provided by circulating APOL1, variants in the pathogens arose which required a countermeasure by humans, and these occurred in sub-Saharan Africa. 2 species of trypanosoma, namely gambiense in western sub-Saharan Africa, rhodesiense in eastern sub-Saharan Africa, which contain in them, in the case of G1, a derived mutant variant, a missense haplotype, only one of which is really important in the association with kidney disease, and the other, G2, for resistance to rhodesiense, is a 2 amino acid deletion, and they're both in if this is the overall N terminus and C terminus, I'm not sure this is working, it's in the carboxy terminus region, which binds a particular region in one of the resistant trypanosome species. So I've talked about this in the past, and having 2 of these derived trypanosome protective alleles in an individual gives them great protection, but at the same time increases tremendously susceptibility to a spectrum of progressive chronic kidney diseases. So that's the realization that led us to continue the work, but all of that was association work, right? And so to try to understand mechanism and to really motivate further study, and this picture comes from Etty, I think Crusoe was sitting there, association that's all circumstantial, maybe compelling circumstantial, so is it enough to really motivate a lot of continued work? And I think, I'm going to show some others, but I think a paper, not from our group, from one of the very active groups now in the field, but recently so, in which the first author is at Tel Aviv, Pazit Beckerman. She is, during her fellowship with Kathleen Chutick in Philadelphia, generated a murine transgenic model with podocyte, and I'll discuss the podocyte in a minute because it's going to be our most important cell for further study, specific inducible expression of either the ancestral non-risk version or the derived kidney risk versions, and in just short, it's a beautiful paper. It really, I think, shows that it's not association, it's causation. That is to say, these mice with inducible expression show a phenotype which is very similar to many forms of human kidney disease that are seen in sub-Saharan Africans, focal segmental glomerulosclerosis, and the whole family of those kidney diseases. So I think that was very, very compelling and important, also can provide a future platform when I get to the drug discovery component for drug testing. And I think particularly encouraging in this study was the fact that this was doxycycline inducible, was the fact that there was a concentration dependence and a reversibility. So even after there was some proteinuria in the mice, stopping or decreasing the expression of the apolipoprotein L1, the human version, which mice have no such gene, was able to reduce the proteinuria and cause regression. So that is perhaps even more important. So the focus is on podocyte-specific expression. In the same study, there was tubule-specific expression, which did not cause kidney disease, so that was an important control. So what are these podocytes? I've spoken about them briefly in past presentations, but they are epithelial-like cells which wrap around the capillaries of the filtering portion of the kidney, the glomeruli, and they are very responsible, very important in the generation of what's called the glomerular filtration barrier, which separates, which allows the kidney to separate the filtration of fluid and small solutes from macromolecules. And many forms of progressive chronic kidney disease originate with a loss of podocyte integrity. They're shed in the urine, they die, and podocytes face the highest endocytic flux of any known cell type. You can think of the filtration process and what they're exposed to, and they're very limited, if at all, in their ability to self-renew. So we're born with a repertoire of podocytes, and they're probably not very much ability to renew them over lifetime. So they're exquisitely susceptible to disruption of endosomal trafficking and autophagy pathways. So here we see again these normal-looking podocytes, and when podocytes die, those that are remaining compensate not by replicating themselves or recruiting something from a stem cell source, but rather they undergo this kind of rather less intelligent kind of reaction, which is to efface their foot processes, hypertrophy, and actually eventually result in decreased filtration and loss of protein. Now, we can grow human podocytes in culture, and many labs have been doing this over the years, and we have to, and you can see that expression of apolipoprotein L1, the G1 and G2 risk variants cause a gain of function injury to podocytes in culture. Many different ways of measuring it. Many have been developed by the people in the lab here, and here we can see in a study in a collaboration with a group in New York, but these are just two of many ways of showing differential gain-of-function injury to podocytes with expression of the G1, G2 risk variants of ApoL1 compared to the non-risk variants, so cathepsin leakage or cytoskeletal disruption. Also now, that was by transient transfection. You can use inducible expression of apolipoprotein L1. Also, you can play with it in a way that it has a secreted version or non-secreted version, and one can show that it's the intracellular gain of function, not a circulating moiety that's hurting the podocytes or other cells, as I'll show you in culture. And with inducible expression of ApoL1 in human podocytes, non-risk and risk variants, there is endoplasmic reticulum localization. I'll show some more of that in a minute. Partial mitochondrial localization, and again, inducible expression of G1 and G2. This is G2, causes a time course for kidney risk variant cytotoxicity of podocytes. This is another, and there's no difference, though, in the localization. It's not a difference in localization between G1, G2, and G0 so far that we can see. And here, this is shown a little more elegantly with apolipoprotein L1 localization with markers of the endoplasmic reticulum, such as calnexin, or SEC61, but not with the cell surface, such as with wheat germ agglutinin. This differential cell injury is also evident with inducible expression of ApoL1 and risk variants, not only in podocytes, but also in many different kinds of cells, such as HEK cells, human embryonic kidney cells, and in other cells and organisms. So we postulate that the ApoL1 kidney risk variants cause a gain of function, leading to injury via pathways which are conserved in eukaryotic cell function, and to which podocytes are particularly sensitive, and that's why that's what the human phenotype is. That doesn't mean we have to limit our studies to podocytes in culture. We will continue to do studies in podocytes in culture, etc., but we can perhaps, and we have been doing studies in other organisms, other cell types, and looking at this gain of function injury, which we think is impinging on conserved pathways, and these are HEK cells, and again, a differential 12 hours and 24 hours gain of injury. That means we can look to a variety, and other labs around the world who are very active in this field have been looking at a variety of different platforms. So as I showed you, I think, in the symposium 2 years ago, we've used flies and yeast, And I'll just summarize what we think, or what many groups together think is going on today in a cartoon style, and then show just one or two pieces of evidence supporting this current view of what the mechanism of gain of injury involves. It's certainly not completely understood, it's not comprehensive, but there's been some progress, and that is that the APOL1 high-risk genotype causes susceptibility, then with a second hit, such as an increase in expression of APOL1 under interferon stimulation, for example, or other second hits, because not all patients at genotypic risk get disease, as I think I showed on, didn't mention, but showed on an earlier slide. The podocyte is particularly susceptible, is injured. That injury involves a peroptotic cell death, at least in the podocyte, with potassium efflux, problems with endocytic trafficking, inhibition and impaired acidification in the endosomal compartments, autophagic flux inhibition, mitochondrial dysfunction. What comes first, what comes second, etc., is really the focus of a lot of study and debate, which we're also involved in as well. Just to show you just a little bit of the evidence that supports some of that, this was a slide that I had showed actually previously, in which one can see that even a Drosophila is a good model to study at least the differential gain of injury toxicity, and it's actually most dramatic with expression of the G1 and G2. This is work in collaboration with Professor Eddie Salzberg's lab, done by Tali Shemer and others in the lab, and you can see that the Drosophila, when APOL1 is expressed very powerfully, under a daughterless driver, doesn't really suffer, but when just the G1 or G2 variants, which are small variants, they cause lethality. Since that time, we focused on the Drosophila kidney. So the Drosophila kidney is very interesting. Just like podocytes, the flies have nephrocytes, and they don't actually generate urine, but they have the same structure of a filtration barrier. The nephrocyte acts like the podocyte and is able to be involved in the uptake of the products of fly metabolism, sequester it within the nephrocyte cell. And so that's interesting, but it also provides, it's a professional endocytic machine that one can monitor. So if we express now APOL1, G0, the non-risk, G1 or G2, so this is the, I'm just trying to get the pointer, okay. So the G0, G1, G2, and these are controls. One can see that these nephrocytes, and now these are flies which have been engineered to constitutively produce and put into their circulation a tracer. This is ANF, RFP. So one can look at the uptake of that tracer, and in the G0 nephrocytes, there is a slight, perhaps, block in the degradation. But in the G1 or G2, there is an almost complete block of the degradation of the tracer, which has been taken up by these professional endocytic cells. And eventually, in the adult fly, the nephrocytes die. So now with flies, one can do a lot of genetic studies. We're also looking at using all the models at our disposal. So again, work done by Allah to look at this endoplasmic reticulum pathway, using a trick, which is a collaboration with Waldemar Popik, to prevent the entry through the translocon of APOL1 into the ER by creating a mutation, MSA-LFL, in the first part of the fourth exon. One can abrogate signal peptide cleavage. Then one can see that the localization of APOL1 is no longer into the ER, and that abrogates toxicity. So that's a potential future target, again, to abrogate APOL1 toxicity when we get to different ways of approaching a therapeutic approach. And that is strengthened by another set of studies done in the lab in yeast by taking a screen now, a deletion screen in S. cerevisiae, which are just like mammalian cells, differentially susceptible to G1 and G2 versus G0, probably through very similar, highly conserved mechanisms. The rescues are those genes which prevent translocation into the ER. So there's obviously a very important role. Another approach to understand the mechanism is to look for protein-target interactions. This is a collaboration with Adi Kimchi at the Weizmann Institute, using her protein fragment complementation assay, and 2 protein-target interactions were discovered very strongly using this approach, and then Ira Bavli, to corroborate this using a completely independent orthogonal approach, was able to show in a proximity ligation assay, whose details I won't go into, but one can see these are the signals here of the same 2 protein-target interactions. This is the method, and, oops, sorry. Back. Where are we? Going the wrong way, I think. Right. And this is these dots here are the signals of the interaction of ApoL1 with each of these 2 putative protein targets whose disruption might be important therapeutically. So that brings me to therapeutic drug discovery, which will be the last part of my presentation. So while everyone's continuing to work on the mechanism, the question is, it may take many years to actually figure it all out. And should we just wait to understand everything we can about the mechanism, the biology, or can we jump ahead and try to use the models at hand for therapeutic drug discovery? So the appeal is great because it would be possible with a safe and specific drug to administer it to those who are at genotypic risk, and that's estimated to be about 80 million people in the world, about 6 million African Americans out of the 42, because it's 1 in 8 genotypic risk frequency. So that could be 20 million people, 80 at genotypic risk, and then with a marker, early marker for progression, that might be 20 million people. We saw perhaps from the mouse model that injury is reversible in an early stage. A specific target, I mentioned in a previous talk that it's dispensable except under specific pathogen attack. There are actually humans who are null for this gene, and most non-human species don't have it. Partial inhibition should be effective. Heterozygotes are much less effective than homozygotes or compound heterozygotes for the risk variant. There is a murine, there's more than one now, murine preclinical model that could be used to test efficacy of potential drugs. And there are orphan indications, because there's a huge spectrum of diseases, some of which are classified under orphan diseases. So how could we approach therapeutic drug discovery? Well, one is mechanism-based, and we're continuing to work on that, but we're not going to hold our breath for it, although I do think that the steps most proximate to ApoL1 and its immediate interactions, rather than downstream amplifiers, might be more amenable to specificity, structure-based approaches, and phenotype screens. So there's been quite a bit of progress just very recently in terms of the structure-based approach, and that requires purifying, generating and purifying recombinant ApoL1, something which has evaded efforts for many, many years. Orly joined our lab, and she's, I think, sitting here, and has, here she is, and has had remarkable success recently, and by being able to generate and purify recombinant ApoL1, one can now look at structure, biophysical properties, try to recapitulate those protein-target interactions in recombinant cell-free systems, such as some of the interactions we see are dimerization of ApoL1, for example. Use DNA-encoded chemical libraries, which is a very robust, very, very high throughput drug discovery method, but that it requires a cell-free system and recombinant protein that's, I'll speak about in a minute. And then when one can identify compounds which bind to, because that's what DECL does, to the target of interest, ApoL1, perhaps differently for G0, G1, or G2, or maybe not, because we would just give it to those who are at genotypic risk, then you also have a tool for another method, that is for a PROTAC method, because the ultimate specificity would be just to destroy ApoL1 itself, which if it's not needed. So we could use this approach then for drug discovery using another approach. So this DECL could facilitate the PROTAC approach, or simply come out, because DECL yields many, many thousands of potential compounds out of many, many millions which are screened in a phenotype screen. Which brings me now, I think I'm going to skip this slide because it is getting late, although there's been, Orly has also done, and the others in the lab, some interesting work on the biophysics of biophysical structure relationship. But at the same time, in collaboration with the services of the Israel National Personalized Medicine Center's Drug Discovery Unit, we've been doing straight phenotype screens in which we use inducible expression of APOL1 initially in HEK cells. Ira Bavli's been working on this in the lab, but also creating podocytes. And one can see with an assay which is highly amenable to high throughput, that is an ATP measurement assay, whose details I won't go into, a very, very lovely platform for being able to screen tens of thousands, up to 100,000, as I'll show you, compounds, and look for rescue of induced, APOL1-induced drug cell injury. And the parameters, this is a quality kind of, so we started with a pre-screen, and then we recently did a primary high-throughput screen with 100,000 compounds. The first full screen yielded 66 rescue hits out of 2,300 bioactive compounds from most of them FDA-approved drugs, which could be potentially repurposed if they're able to be used in this way, and 560 rescue hits out of 95,000 chemical diversity library compounds. And then on a validation round, this narrowed down to 125 compounds. So that's a tractable number that one can start working with medicinal chemistry and so forth. So this leads me right towards the end. So the approach has been mechanism-based, phenotype-based compound screen, now cell-free recombinant DNA with biophysical assays that one can look at, protein-protein interactions, and these more recent drug discovery approaches that I alluded to, combining with medicinal chemistry. And ultimately, one would need to go, and because there are murine models, I think we're really, and many other groups are very, very interested in this, in progressing towards drug discovery, in parallel working through mechanism. So I've tried to really give a snapshot of quite a bit of work. A lot of it is collaborative, and it involves an interactome. This is part of the lab interactome. So we have people, many of them are sitting here, and these are only some of them in this picture, who are currently working on the project, who've recently joined the project and working on other things, who've been in the project. I would like to be able to name them all, but they're here, and I'm very, very deeply grateful for the privilege of working with them. Collaborators at the Technion, such as Adi Salzberg, Daniel Kornitzer, now Yoni Savir. Others in the lab are working on other projects, sitting in the audience, who come to the lab meetings and always have something very important to contribute. Collaborating PIs at other institutions in Israel and abroad, in the United States, in Europe, in Africa, because I haven't shown some of the genetic epidemiology work for lack of time. And this is probably the most gratifying part, is being able to work with so many excellent people. So we're in the lab a lot, but we also go out every so often. And this is, I think, the second time the lab went out to the Hula Valley and for a trip recently. And I can tell you the only thing that the people in the lab allow me to do is a little bit of work, which is, let's see if I can show it to you here. There it is. All they allow me to do is, drive the little bicycle during the trip. Otherwise, they won't let me touch a pipette or anything because they want the results to be good and accurate. So again, I'd like to express my thanks to the program, to everyone involved in it, especially to people in the lab and to the Beutler family and to Bruce, and I thank you very much for your attention.